Okay, so this week we're going to start our unit on chromosomes. This is unit 3.2. And before we, dive into that, first of all, what is a chromosome? So last week we dove deep into the structure of DNA. So we know that DNA is a polymer of these nucleotide monomers. So this would be one DNA polymer. So polymer just means chain. This would be another DNA polymer. Again, these nucleotides, those are your monomers. Remember, they're made of the phosphate that's covalently linked to your sugar, which is covalently linked to your base. And that DNA forms these base pairs, which are linked together by hydrogen bonds. And remember that G always pairs with C and A always pairs with T. So if we think back to even the first week, we learned that your genes are encoded by your DNA. So if I were to look at a DNA molecule, maybe one gene is encoded here and another gene is encoded on this part of the DNA molecule. And remember that DNA does more than just code for protein coding genes. You also have those regulatory sequences like your promoters, like your exons and your introns. So we learned all that in week 1. But DNA doesn't exist just in a free form like this. It's actually condensed into a really specific structure called a chromosome, and they look something like this. So it's all wound up. The DNA molecules are wound up in these structures. And when you zoom out into a cell and you look at them under a microscope, you might have seen these sort of thread-like structures under the cells that you were looking at in the lab last week. And those are chromosomes. They're just DNA that's wound up really tightly. Okay. So this is going to go in your guided notes. The official definition of a chromosome is that it's a thread-like structure made up of condensed molecule DNA containing a specific sequence of genes, but also their regulatory sequences. And again, this is what they look like, these sort of thread-like structures. These are the chromosomes, so they're wound up molecules of DNA. So not all chromosomes are created equally. It depends on what sort of cell type that you're looking at. So there are 2 main classes of living things in biology that you have to be aware of. Prokaryotes, which are bacteria, and this is a picture of them here. And then we also have eukaryotes, and these are what we are. So this is a picture of a human cell, human cheek cells, like the ones that you were seeing in your lab. And this is a picture of some plant cells. And what you probably noticed right away is that there is a difference in structure here, where eukaryotes have this circular structure, which is the nucleus. Prokaryotes don't have that. So eukaryotes have a nucleus, which contains the DNA. So the DNA in the plant cell is here. When you were looking at your cheek cells, you had sort of this larger, lighter blue area, and then in the center there was a dark blue dot. That's your nucleus, and that is where your DNA is. So we are eukaryotes, so we have a nucleus. And prokaryotes, and like you can see over here, there's no real center in this molecule. The DNA is just free in the cell. So, okay, again, those are the nucleus. This is the nucleus here in this cell, no nucleus here in bacteria that you can see. Okay, so all living things store DNA in chromosomes. Again, that genetic code is universal. It'll work if I take a gene in a human and put it in a bacteria, it's going to work there. But the chromosomes are not stored in the same way. So again, this is a picture of a real bacteria, and all prokaryotes are bacteria. And here we have on the right side, I think this is like an onion cell. So this is a eukaryote. And you can see that there's a pretty big difference here. So this is the chromosome in the center of this prokaryote. And these are the chromosomes over here in the eukaryotes. So you can see that they are a couple differences here. One, this one is more disorganized. And 2, there seem to be multiple molecules over here. So let's go talk some more about the differences between prokaryotes. So each of these have a chromosome. So all living things have a chromosome, but they have different structures, right? So this is a model. On the left here, we have a prokaryotic, let's say a bacterial cell. And then on the right, again, I have a model of a eukaryotic cell. Again, eukaryotes have this nucleus and prokaryotes don't. So just from looking at this, if I told you that these were the chromosomes, you can tell that there's a couple differences here, right? So this one is circular, and there's only one. And then over here, we have multiple chromosomes, and they have a different shape. They're not circular, they're linear. So in your notes, I want you to know that within both prokaryotes and eukaryotes, that DNA, it is condensed and stored in the form of chromosomes. These are each chromosomes, but how those chromosomes look is different. And we'll just walk through this one at a time. So for the prokaryotic cell, so these are your bacteria, there is a single chromosome. So that's just a single DNA molecule that's wound up, and it's in the shape of a circle in prokaryotes. So this is different in eukaryotes. So in the prokaryotic, it's a single chromosome, and it's in the shape of a circle. The other thing about prokaryotic chromosomes is they do not contain protein. They only have DNA. So prokaryotic chromosomes do not carry protein, they only have DNA. So again, in your notes, you'd want to have that prokaryotes have this single chromosome that consists of a single strand of this circular DNA. And again, it contains no protein, only DNA. And again, prokaryotes, they do not have a nucleus. So this is located in an area called a nucleoid. You don't need to know that specifically. So just know that single chromosome, circular in shape, contains no protein, and it only has DNA. So prokaryotes also have this unique feature, and then they have these extra pieces of DNA called plasmids. So they're much smaller than a regular chromosome, and you'll just see them hanging out in other parts of the cell. So this is what they sort of look like, these plasmids. Again, all bacterial chromosomes are circular, so these would be small circles of DNA. So these guys can travel really easily between different bacteria. They're one of the ways that antibiotic resistance can be transferred. So again, some prokaryotes contain these smaller circles of DNA called plasmids. You do not find these in eukaryotes. They are only in prokaryotes. And again, it can carry traits like this, antibiotic resistance, and they are transferred through this process called conjugation. Okay. So that's sort of the 411 on prokaryotic chromosomes. So if we look again at our eukaryotic cell, probably again the first things that you're noticing is that there's more than one of them and that they have a really different shape. But let's go ahead and take that closer look. So chromosomes in the eukaryote. Again, first thing is that instead of being circular, they are linear. And again, there's more than one of them. In bacteria and prokaryotes, there's only one, and it's a circle. And in chromosomes, it is linear, and there's more than one. And in contrast to prokaryotic cells, the chromosomes of eukaryotes contain both DNA that is associated with a histone protein. So prokaryotes, no protein, just DNA, and in eukaryotes they have both DNA that is associated with protein. So again, the other thing that you're seeing here is that eukaryotes have multiple chromosomes, and each of these chromosomes contain the different genes, right? So if this is, I think, a karyogram of humans, so on this chromosome, you might have a gene for, say, hair color. And on this different chromosome over here, you might have the gene for the PTC detection. And on a different chromosome somewhere else, you might have a height gene. So in a eukaryotic cell, you'd have these multiple linear chromosomes, but each of them contains different genes. Okay? So eukaryotes have 2 or more chromosomes, with each carrying a different set of genes. So this chromosome is going to have a different set of genes than that chromosome. It's going to have a different set of genes than that chromosome. You can think of them as like volumes of the human code. So this would be book 1, this would be book 2, this would be book 3, and each of those books have different recipes for the different proteins that we use. And each chromosome is going to have a different set of genes. Okay? So let's look at this last point here, that eukaryotic DNA contains both, or eukaryotic chromosomes contain both DNA and histone protein. So it's packaged very specifically. So if we have, this is our eukaryotic DNA, and it's just hanging out here, and it's about to attach to a protein called a histone. There it goes there. So this histone protein grabs onto that DNA, and it's going to wrap the DNA around itself, forming these sort of string bead structures. and that structure is called a nucleosome. I don't think you guys need that much detail yet, but they do keep forming these higher order structures into these chromatin fibers. And this is how eukaryotic DNA is assembled. It's these DNA protein complexes that just keep sort of winding over and over onto themselves. And what you're eventually going to see here is that it's going to keep packaging until it forms that final chromosome shape that you've been familiar with, those linear chromosomal molecules. Okay, so again, this is just the big fibers, and it's going to zoom out here in a minute. So this is just a really super wound up piece of DNA in the chromosome. Okay, and again, if we zoom out, this would be what they look like in the nucleus of a eukaryotic cell. Okay, and you can only see them when cells are dividing. So for your notes, you're going to want to know that unlike prokaryotes, DNA in eukaryotes is associated with this histone protein. So again, here we have our double helix molecule of DNA, and it's going to just wrap around these histone proteins. And this is different than prokaryotes. Prokaryotes don't do this, it's just straight DNA. But in eukaryotes, the DNA wraps around the histone proteins. This is going to allow, like you saw in that video, the molecules to wrap around these proteins and condense. And you guys have 6 feet of DNA in your cells right now, and it allows them to be stored in a very compact matter. And that is called supercoiling, because it's hypercoiled. And again, this is just another visual of what that looks like. Again, we have this DNA, it's wrapping around the nucleosomes, the histone proteins here, and it just keeps stacking until it is wrapped very tightly. Okay, so if we can then compare chromosomes in prokaryotes and eukaryotes, so what they both have in common is that they both have at least one chromosome of DNA. So that is true whether you're bacteria or you're anything else. So prokaryotic and eukaryotic cells both have chromosomes made of DNA. Remember, that genetic code is universal. But there are some differences that we've covered. So again, in eukaryotes, what sets them apart is that they have a nucleus. Again, these are the little plant cells. You can see the big circle in the middle, and that nucleus is where the chromosomes, where the DNA is. Where prokaryotes do not have that nucleus. You can see there's an absence of that nucleus structure in prokaryotes. Again, prokaryotes have a circular chromosome, whereas in eukaryotes, they are linear, they're straight lines. And in prokaryotic cells, there's only ever one. So prokaryotes only have a single circular chromosome, whereas eukaryotes have multiple linear chromosomes, okay? And the other thing is that prokaryotes are unique in that they have plasmids. So those are the little extracellular circles of DNA. So only prokaryotes have plasmids. Eukaryotes do not have plasmids. And again, the prokaryote cell does not contain any protein. It's just, they'll sometimes call it naked DNA. So there's no protein associated with the chromosomal DNA in prokaryotes. But in eukaryotes, they do have that histone protein that is helping to wrap the DNA into those chromosomal structures. So that's the crash course in prokaryotic and eukaryotic chromosomes, and that will be what we are covering for day 1. All right.